Hi all. Thank you for attending our webinar, Should We Change Chimeric Human-Animal Research? We will get started in a few minutes, waiting for more people to join us. Hi all, thank you for joining our webinar today. We will start in a few minutes, just waiting on a few more people to join in. Hi everyone, thank you for attending our webinar, Should We Change Chimeric Human-Animal Research? Today you will not be visible or audible during the webinar, but we do encourage you to ask questions through the Q&A function. The session is being recorded and it will be available at the Hastings Center website later today for viewing with closed captioning. This webinar will focus on the recommendations recently addressed in the Hastings Center Special Report, Creating Chimeric Animals, Seeking Clarity on Ethics and Oversight. It's now my pleasure to introduce today's speakers. We will be joined by Josephine Johnston and Dr. Insoo Hyun. Josephine Johnston is a research scholar at the Hastings Center and is an expert on the ethical, legal, and policy implications of biomedical technologies. She is also a lecturer at the University of Otago. Dr. Hyun's work focuses on ethical and policy issues in stem cell research and new biotechnologies, and he is the inaugural director of the Center for Life Sciences and Public Learning at the Museum of Science in Boston. Dr. Hyun is also a member of the Center of Bioethics at Harvard Medical School.
Morena, tēnā koutou, tēnā koutou, tēnā koutou katoa. Welcome and thank you, Danny, for that kind introduction. Thank you to everyone who's online now, zooming in from wherever you're zooming in from around the world, maybe. I'm zooming in from the other side of the world myself, just full disclosure. So thanks for joining us for this conversation about the ethics and oversight of human, non-human animal chimera research. In just a minute, we will say a little bit more about what that is, what we mean when we say that. But let me first of all acknowledge that a lot of what we're going to say today draws on a NIH-funded research project that my colleague Karen Maschke, Insoo, and I led with working collaboratively with Patricia Marshall at Case Western and Carolyn Newhouse at the Hastings Center, and with the very amazing support and involvement of Caitlin Craig, Meg Matthews, Isabel Bolo, and Ben Wills, and drawing on the expertise of an international interdisciplinary work group that we put together. And many of the findings of the project are in the November-December Hastings Center special report that you can access, open access, on the Hastings Center's website and through the Wiley platform. So this project that Insoo, Karen and I co-led had various components, and we will draw today, Insoo and I, on the qualitative analyses that we did, or interviews and content analysis, and on the conceptual, normative and policy analysis we did, especially with the work group. But Insoo and I also have our own histories of looking at and researching and playing roles related to this kind of research. So Insoo's been a member of and a leader at the International Society for Stem Cell Research for quite a long time. He'll say more about that, which I know informs his thinking and his understanding of these issues. And I've been following and writing about the stem cell debate, as has Insoo, sort of since 1998 or 9 or something, when it all kind of took off. And that's very much related to the history of this issue from a policy perspective. I also served on an embryonic stem cell research oversight committee, which we'll say what that is in a second, but that looked at studies, including ones that involve the creation of chimeric animals. And so, we're going to draw on lots of things, including our study, but not just that in our discussion today. Okay, well, before I go much further, I just wanted to ask, I'm going to ask Insoo if he can give us a sort of working definition of chimeric research so that we can, sort of all be on the same page from the beginning.
Yeah, great. Thank you, Josie, and thank you, Danny, for the introduction. It's great to be here. I believe also along the way, when you have questions that come up, our participants can ask questions. I'm not really sure if it's a Q&A function or the chat, but maybe Danny can put up into the chat instructions for how to submit your questions. We'll be happy to take those later on. So chimera research, as Josie said, has been something that we've been thinking about for quite some time. I don't know how many of you in the audience are really all that familiar with chimera research. So I just wanted to say a few brief things about what chimeras are and why they're being used for research. First, what are chimeras? Chimeras are organisms that are composed of cells that come from at least two different zygotes. So that's like the most general definition of a chimera. Now, to get a little bit more specific, these two zygotes could be of the same species or different species. In nature, sometimes you get embryos that fuse in the womb very early on in development, and when the embryos fuse together and survive, they will form a species to species, same species, chimera, which could be a combination of a male and female zygote or both of the same sex. But in these cases, you have one organism that's composed of cells that are side by side, genetically distinct. Okay, now that's different than creating an organism through breeding of two different species. That's what we call a hybrid. A hybrid is when you mix sperm and egg from two different species, and then the mixture appears at the genetic level within every single cell in that animal. Okay, so a mule, right, et cetera, et cetera. So we're not talking about hybrids, because to make a chimera for research, what you would then have to do, a hybrid for research, what you would have to do is you'd have to combine sperm and egg, and that's not the kind of experiment we're talking about. So it's any animal that has cells that have integrated from another zygote. And like I said, the most common ones are same species. In research, in stem cell research early on, most of the chimeras were mouse chimeras from two different strains. So they would take, to prove that mouse embryonic stem cells were truly powerful, researchers would inject embryonic stem cells from one zygote, from one blastocyst of, let's say, a white-coated mouse, into the blastocyst of a brown-coated mouse, a different zygote. And if that integrated, then you would get a mouse that was born that had patches of brown and white, which meant then that these cells didn't integrate at the nuclear level throughout the body, but rather you had white-coated cells next to brown-coated cells, and they existed side by side. So those are the same species ones. In the human case, we do have clinical chimeras. We don't call them that. We call them the transplant recipients, right? But you get, somebody who gets a donor heart from clearly a different zygote, a different human being, and that's transplanted into clinically into the recipient. So that person would have either male or female, right, solid organ in their body, and they would be technically a chimera, one organism composed of cells that come from different zygotes. But again, nobody really fears the human-to-human chimera. I guess there are also cases where people, individuals with fuse in the womb during development. So you might have some forms of hermaphrodites might be from that. You can get cases where people did, for whatever reason, a genetic blood test from blood and compared it to from the gut, and you get two different genetic readings, which is crazy, but that meant that you had an absorbed twin in the body at some point. So those are naturally occurring chimeras or clinical chimeras. The chimeras that Josie and I are going to talk with you folks about are research chimeras, and really not the mouse-to-mouse studies that I mentioned, or even the rat-to-mouse studies, although that might come up later. We're talking about human-to-animal chimeras. So the donor cells come from human, and they're transplanted into a research laboratory animal and allowed to integrate and go on from there. Now, I do not mean to suggest, we do not mean to suggest that chimera research is really that new. It's been around for decades. The stem cell chimera work is relatively new, but other kinds of chimera work where, you would transplant human tumor cells into an immune deficient mouse, for example, or even you might supplant thymus tissue into a newborn mouse to create what's called a human immune system model within a mouse. There have been lots of examples from, blood studies, leukemia studies, cancer studies, where people have mixed human donor materials into an animal and had it integrate. I think the whole issue of the stem cell chimera, really took the possibilities of chimerism theoretically to a much higher level than in the past, right? So in the past, people didn't really worry too much, and still today don't worry too much about very limited chimerism where you have, again, in an immune deficient animal, the transfer of human cells to biologically humanize the animal in some way to study it. But with stem cells, there's the possibility, theoretically, that the chimerism could be much more severe, much more acute. And the reason for that is the stem cells are very powerful cells, and you can put them at various stages of development. You can put it, as I suggested, in the pre-implantation stage of an animal embryo. You can put them, in utero, within, the developing fetal animal, or postnatal. So basically, there's a whole continuum of different kinds of research chimeras using stem cells. And the variables are, what are the species of the host animal? What is the developmental stage of the host animal? How many human stem cells are you putting in? Where are you putting them in? How long are you letting them develop? And for what purpose? So those are the broad array of activities. Maybe I'll turn it over to you, Josie. I mean, there's certainly issues that come up. Like, people ask questions like, well, why would you even make a chimera for research? Maybe we can kind of get into some of that.
Yeah.
We've heard that all the time. Is it just curiosity, or, why would anybody want to put human, you might understand the cancer cells. Yeah, you want to make the mouse into a little human cancer patient and test your drugs, but why would you put human stem cells into an animal? I was wondering if you ever got that kind of question, or, people wondering out loud to you, why are people even making these?
Yeah, well, I think people's hope I don't know what people intuitively would think, but certainly the way that I've been thinking about it is that there are sort of two main drivers for creating either in a dish, an organism like a blastocyst that has a mixture of human and non-human animal cells, or actually having that happen in a living animal. And it seems like they're either primarily about modeling, so creating models of human disease. And if you want to model some human diseases, you might want quite an integrated, the human cells integrated in quite a significant way in the living animal to model the disease, or in the future to be able to sort of help with regenerative medicine. So be able to have a pig that has got a human kidney living in it, and that would be the kind of output of chimeric studies. So I think one of the distinctions, so that's a distinction that I found helpful, thinking about modeling of human disease and creating, hopefully, or trying to create more accurate models for things that we can't easily model in living, just by studying humans, or by studying animals that haven't been modified. And then the other main purpose being in order to sort of move towards various kinds of regenerative medicine goals. The other distinction that I think is quite helpful is thinking about chimeric research as being done in a lab, in a dish. There is no living chimeric animal. You still probably have had to use animals to get to that point, whether it's by taking sperm or eggs, or some other, or some other cells from animals, non-human animals. So it's not that non-human animals aren't involved, but what you're studying is not a living animal, or and distinguishing that a little bit from in vivo or living animals that we are either creating as part of these studies. So I do think that that's a distinction that I've also found quite useful in this sort of just laying the landscape of this debate out a bit.
Right, right. Yeah, I'm glad you pointed that difference out. I did so much of my work within the context of the International Society for Stem Cell Research, where I was the ethics chairperson and I helped draft all their guidelines. And within that scientific community, somebody like me coming from philosophy and bioethics, who sort of was learning the science sort of on a need to know basis along the way, very quickly realized this is such an enormously broad area of research. I think a lot of people, when they think about chimera research and the controversies around it, they have something kind of particular in mind. But I think what people need to realize is it's extremely broad, and there could be some really quite different reasons why a research team might put human cells into an animal model at some stage of development. So I think a really interesting category just to kind of think about is the animal host pre-implantation, right? Is it all done in a dish? So from a scientific point of view, or even from an oversight, like regulatory point of view, that's sort of not viewed, surprisingly, maybe to some people, not viewed as like super contentious or controversial or hot button, because it's not using what's thought to be, like a living animal with a central nervous system for which you need an animal oversight committee there to monitor pain and suffering, et cetera. Of course, there may be animal monitoring if you're clearly procuring the gametes, the sperm and eggs, to make the little embryo to then put the stem cells in. If you're procuring those from living animals, of course, you're going to have some animal welfare, just looking at the welfare of the gamete donors. But once you have the gametes in the lab and you combine them in a dish to make your pre-implantation embryo, so basically animal IVF, when you've done that, and all you're doing is just keeping it in the dish. So even after you put the human cells in there, if you're just keeping it in the dish, from a regulatory point of view, that's not really like that big of a deal. But for many people, because it's so early in the animal's development, and depending on the species of the embryo you're talking about, whether it's mouse, which doesn't work very well at all, or something closer to human, like a non-human primate, people do get a little bit worked up about these experiments. It really does start to kind of like seem like you're really crossing a line there somehow. But from kind of a general everyday regulatory point of view, it's not that hot button, because it doesn't seem to trigger like the normal red flags that come up in review, like pain and suffering, because the thing doesn't even have like nerve cells at that point, right? The little embryo doesn't. So one big category is kind of like in the dish, it's going to stay in the dish. Well, we realized that the ISSCR, that's the International Society for Stem Cell Research, is things get a little more complicated than when you transfer it into the womb of a surrogate animal, and then you gestate that mixture, the mixed embryo, for some time. And there could even be theoretically all the way to gestation, full gestation and delivery or birth of the animal. So there are like these various stages. You almost like think like, what are the different stages of development of any animal? And at any time point, you might imagine cells going in. And then the other big like hot button issue for people is not just human cells in the animal, but once the animal is like, is gestated and birthed, if it's postnatal, if the cells go in, if the human cells go into the central nervous system, that tends to be a lot more like red flag waving than just like, let's say, human pancreatic cells put into a pig or put into, another animal, just to see how well the cells survive in a living context. Like those kinds of like non-neuronal cells tend to be less red, but like hot button. And the closer you get to the brain or the sex cells, the more kind of hot button, I guess. I love one time Hank Greeley, which many of you know, he said it's brains and gonads that sort of get people all worked up. Right? I think he's right when it comes to chimeras. It's the brain and anything involving sex cells that kind of really raise the issue.
Yeah.
I would add to that the embryo also, if you're putting it in the embryo.
Yeah. So actually, I think maybe that's a good shift over to sort of like what some of the ethics and oversight issues are in this area. And maybe I can just like kind of explain that, like maybe like some other people on the Zoom, I came into this via the embryonic stem cell controversy. And so, in 1998, or I think it was, there was the first derivation of embryonic stem cells from, derivation of these pluripotent stem cells from human embryos. And I was just starting my career in bioethics, actually doing a master's of bioethics. And, I wrote a paper about like a scoping paper for the New Zealand government, actually, about what sort of countries around the world were doing in terms of regulation and oversight of this area of science. And that was very focused on the fact that we were, the science involves taking pluripotent stem cells out of human embryos. Mostly those embryos have been created in the course of IVF by patients who then don't want to donate them to anyone else, but also don't want to use them themselves, or have finished having their family, or have just finished with IVF. And that the embryo is therefore destroyed. And in, various countries have very different reactions to that. But as we know, in the US, anything involving embryos has a long history of being very controversial. And even before the derivation of embryonic stem cells, there was already a law limiting the amount, the use of federal funds for any research involving sort of harm to or destruction of human embryos. So this research fell into that and hit up against all of those abortion and embryo-related issues in America. So that's kind of how I started thinking about it. And then over the course of, I think many people, know this, of course, that like 1998, there's the derivation, 2001, there's the President George W. Bush's pretty well-known policy, putting pretty significant limits on the funding of research with embryonic stem cells with federal money, then private donors and states, including, quite notably California through a referendum with its voters, step in. And it's a very interesting political and science kind of mix there of states and private founders stepping in and funding the research. And then you have this thing where researchers would have sort of like one set of equipment for federally funded research, and then a completely separate sort of setup for the non-federally funded embryonic stem cell research. And already in that was this possibility, as a kind of subset of the embryonic stem cell debate, this possibility that embryonic stem cells would be mixed with, from humans, would be mixed with non-human animal cells. And so in 2005, when the National Academies of Science group put out its first set of guidelines for this work, sort of stepping in to provide guidelines for research that the federal government was not funding, therefore NIH didn't have guidelines for. So they put guidelines out. They already had a subset of those that were addressing chimeric studies. And as you said, those guidelines in their various iterations, and then ISCAR's guidelines, when those started to come out, was it first in 2012, I think? Is that right? Or 20?
So when we did touch on chimera research, it was actually all the way back in 2006, the first
2006.
A little bit.
Okay. Yeah, so those guidelines all had something about chimeric research, and those studies, the guidelines focused on studies where, the concern, I guess, that there might be human gametes produced by these non-human animals. So like you said, gonads, so nobody wanted, and the usual mechanism for that was like the animals can't breed, but wouldn't really explain why. But behind that, presumably, is this concern that there might be human gametes produced. And then you would get in that case, a hybrid. It would actually, right, if it worked. So, and then here, the other big sort of focus would be on like those studies in which there was a significant possibility that, or a possibility that significant contribution to the non-human animal's nervous system or brain would be part of the study. And what that meant for oversight committees that were at various different institutions looking at this research is that you would, and I know this from experience, you'd be sort of asking scientists to tell you things like, do you think that the animal is going to produce human sperm? Or do you think that the animal is going to have a significant portion of its brain with human cells? And it sometimes felt like we were asking sort of very sci-fi questions and not really having a lot of background behind us of like, what's this really about? What's the main ethical concern driving this? Something probably about like, what would it mean for humans to have these creatures out there that had something like human brains to some degree? But I felt like it was a rather inchoate discussion. That's right. And that's sort of part of the motivation for our project, was to try to dig in a little bit in this stuff and sort of try to clarify, like, what do we think is mainly at stake here? How can we express it? What kinds of oversight makes sense? What's already being done? And so that's sort of where we started to dig in our project.
I think you raised some really interesting points. I think it's crucial that the audience think about the actual timeline and how long some of these concerns have been around and how some of them just did not stand up to the test of time. I remember in the early days of this debate, there were some people, this is back in the George W. Bush era, to like 2005, 2006, 2007, around there, where there were even like bioethicists, I'm not going to say who, but somebody on George W. Bush's Bioethics Council who raised like some really provocative concerns, but really were kind of scientifically nonsensical. So one person said, I would be very concerned if somebody made a sheep with a human face. It's all right. Well, what would be the scientific reason for doing that? And by the way, how would that even be possible? Because a human face on a sheep would have to involve technically the mesoderm, ectoderm, endoderm, all three germ layers, but only on the face. Like, how would you actually make a chimera model that could do that? So I don't even think it's scientifically possible to do that. So anyway, it was startling to think about something like that happening, or a pig with human hands again. And it involves technically all these germ layers, and you can't get that result using the transfer of human stem cells necessarily. So that was like a very provocative kind of thing to be worried about, right? But it did not stand the test of time for a number of reasons. One, it was just ill-formed developmental biology. But I think another key aspect is people have found over the past few decades, it is really hard to get human cells to survive, and human stem cells even to survive and to integrate and even like grow or do anything in a foreign animal host. And so like even to get like more than like 3%, it's like a really big deal. And the scenarios people were thinking about were like close to like 30% or 50% human. So it's just not like the science isn't there yet, which brings up a really interesting point about, so now that people know there's a very high xeno barrier between human and non-human species, how would you actually overcome that? And that gets us to sort of like why people are doing the embryo work. Okay, so some of you may have heard about some recent experiments, maybe a year or two ago, where they got monkey blastocysts and they put human stem cells in them, and then they found that some of them kind of, integrated for a little while. You got a little bit of cell survival. There are a couple of, so the reason for that is not that they want to eventually grow human organs in monkeys, right, for transplantation, but it's to better understand this barrier between human and non-human, to then better inform livestock animal models that have human stem cells put into them. But that, again, just kind of in the dish system, the rationale for doing some of that work with non-human primate embryos is to better understand how to make the cells survive in other species. So you kind of need to see a little bit more of like what the context is of why people did that kind of work. But a key development that also happened from like 2005 to now is the discovery, the suspicion, later confirmed, that human embryonic stem cells, the ones that come from the embryos from fertility clinics, are not kind of at the same developmental power level as mouse embryonic stem cells. So if you get mouse IVF, and you get a stem cell line from a mouse blastocyst, that mouse pluripotent embryonic stem cell is so much more powerful than what you get from an embryo from a human stem cell infertility clinic. And it was quite different. I mean, they're both called pluripotent stem cells, but the human version is kind of what we would think in the mouse is the epiblast cell. This is like after it's implanted, and it's kind of already going down one of the three germ layers, endoderm, ectoderm, mesoderm. The human version is kind of a little bit further along in development. So the key development since then was to drive it back to about equal to the mouse power, the powerful stem cell. And it's what we call naive stem cells or ground state stem cells. You actually have to do something further to the human embryonic stem cells, or now the iPS cells that you can make from skin, to make them even more basic. And so you kind of get them a little bit more developmentally on par with the mouse embryonic stem cells. And because we now use naive stem cells, iPS or human embryonic stem cells, that better enabled some of the chimeric work to go forward and actually get a little bit more success, but there's still a long ways to go. I remember back when, all the heat and controversy around human embryonic stem cells kind of shifted over into the chimera work, because obviously you're using like these very precious cells that have come from very controversial biomaterials in the human embryo debate. And I remember when the iPS cell revolution came out, this is, the ability to make embryonic like stem cells from any human cell, to reprogram it to do that. I remember thinking, they're going to start using those in animal studies. And there you have potentially living donors who donated like a biopsy. And maybe you have to kind of ask them, are you okay with your cell line being used to make neurons and being put into the animal? So there was that whole other complication you didn't have with the embryonic stem cells, because the embryo was destroyed in the process of getting the cells, but not for the adult or the pediatric patient that provide the cells that are now making these like biologically humanized animal models. So I thought that was very interesting to kind of have that addition to the iPS cell work. But I remember also thinking at that time, even if people supported human embryonic stem cell research, so they were not part of the group that thought it was wrong to derive human embryonic stem cells, I thought there are people out there in the United States and elsewhere that care more about animals than they do about human embryos in an IVF clinic. Like they would place the moral status of animals way higher than an embryo before it's been implanted that's sitting in a dish. So I thought, just you wait, even the supporters of human ES cell research might, when they come to find out that during the course of research, a cell line might be transferred over to an animal, that's going to be a whole other generation of people involved in this debate. So we're, I think, seeing some of that now, right? So the iPS cell revolution didn't side-skirt that issue, actually brought in much more human biomaterial into the chimera realm.
Yeah, so I mean, that's really my experience with this issue, and especially doing this project. And I really have to thank Carolyn Newhouse for sort of waking me up pretty early on. It's like I came in from the stem cell debate and discussions about human embryos, and then like, what does it mean for humans if we create non-human animals that have some of our cells in their brains? Does that mean we owe them something different? And I think some of those issues are definitely here, but one of the things that I think, our project really emphasizes in its recommendations and analysis that we published in the special report is that, like, this is often non-human animal research. Non-human animal welfare is at stake here. The changes and societal shifts in how we think about non-human animals and about non-human animal research, changes including some movements away from primate research, the secrecy sometimes, or the challenges associated with discussing non-human animal research, those are massively at play in this area. And those of us in bioethics have not really, with exceptions, but not really focused on that. We've been mostly focused on, I take myself in this, on questions about these sort of moral status questions, and also those donor questions of like, do the embryo donors know what they're donating to? If they're iPS cells, do the cell donors know, understand what they're donating to? Have we got the right consents? Those are important, but those human-centric concerns, very distinct in a way from these animal-centric concerns that are like, these are non-human animals we're creating and using in this study. We don't want to be talking about this in a way that inadvertently ignores that. And in our report, we really elevate that issue and draw more attention to it, and note that, like, it would be a tragedy if by focusing on these other issues, we sort of inadvertently don't notice that, like, this is an area of non-human animal research. There could be novel forms of suffering that come to the non-human animals as a result of being in these studies. That's a big thing to be looking for related to oversight. And so really kind of trying to put the spotlight more in that area, and noting also that, like, chimeric studies, these will intersect with ongoing larger debates about non-human animals, non-human animal research, court cases about the legal status of different non-human animals. There's very, good, you can imagine those intersecting very publicly with chimeric studies. So I felt like that was a big shift in my own thinking as a result of doing this work.
Yeah, that's terrific. I'll also kind of give a little bit of a confessional to you. I know we're going to turn to questions in a moment, but back when I was at Case Western Reserve University School of Medicine for many years, one of the things that I did very early on as a junior faculty member was I was assigned to serve on the IACUC, the Animal Use and Care Committee, so the animal IRB. And so at that point, I was a young faculty member. I didn't really want to do it. I thought this is kind of, I guess I have to do it because I'm low person on the totem pole. They told me you got to be on this committee. But I was so glad I was on it, because although in the very beginning, it took me quite a while to kind of catch on to like what's going on, how do you actually review animal protocols? I come from philosophy. I didn't even, do any experiments as an undergrad. It was so interesting to see kind of that world. Okay, so the animal care and use committees will go through a whole checklist at the beginning of kind of like how many animals are using, what's the species, why are you using that species and not one that's quote unquote lower on kind of like the scale of complexity, and for how long are you going to use the animals, and what's the pain category. Like there are all these really specific questions that you ask along the way. And when I started then working on the Chimera issue for the International Society, I kind of thought, having had that experience, thought, well, what else are we supposed to be looking for? Because our guidelines, I have to admit, we just said a special escrow committee has to look at Chimera experiments using human stem cells. And I thought, well, what are we supposed to do that's different from the IACUC? And that was really kind of a question that kept like sort of haunting me. I kind of thought, well, we don't want to just do exactly what the IACUC did, because that's what we know what they're supposed to do. What are we supposed to look at? And I realized a lot of people's concerns about Chimera research, to me, initially just seemed like they were just concerns about animal research. They're just the use of animals. If your concern is not that, is there anything extra that you're worried about, anything unique about Chimera research? And that's when we kind of got into the whole like dignity issues, or like, it's not pain and suffering, but it's gain of function. And it's kind of like, getting closer to human and its ambiguous moral status. These are definitely not things that are on that IACUC checklist. I'll tell you that there's no box that says, is there a dignity issue here? There, nobody discusses in committee whether or not there's, like enhanced cognition and therefore maybe a benefit or elevation of the animal's capabilities related to moral status. Like no mention of moral status at all, just pain, just, the animals like agitation, environmental enrichment, those kinds of basic things. So it's really interesting to think, well, what else will we be talking about when we're talking about the ethics of Chimera research that's not simply reducible to animal welfare issues? And I think our project really kind of brought that out even further, that the need to like say, well, if there's really nothing else there realistically at this point in the science to mull over, then maybe really the key issue is animal welfare. And I think that was a really, for me, a good place to land, because I think it did kind of institutionally kind of give people a handle on, well, okay, we kind of already do this. Is there any additional help we need, or is there a reason to kind of be monitoring the horizon to see if anything new might come up that we need a little tweaked approach? And I think that kind of grounding of like, okay, no matter what you think about animal research, here's how it normally is covered. And then trying to like ask like, what else is there? That was a very fruitful way to frame everything. And I think that's what we tried to do in the special report.
Yeah, I think that one of the sort of realities of some of this research is that there's actually a lot of oversight happening at research institutions, at universities and other research institutions from different committees, and it can be a bit siloed. And so like trying to make sure that it's, out, and that the ethics discussion is also a bit siloed. And so we've tried to kind of like bridge that. I just want to quickly show a little bit. I need to show a couple of things related to the project, and then we're going to turn to the Q&A. So let me just explain a little bit about the project. As I said, we were looking at what was ethically at stake in Chimera research, trying to be really clear about what language best captures those concerns. There are a lot of terms, like even chimera, that's like, actually, if you look it up, a kind of a monster in mythology that we thought was, we in our just a preview find isn't helpful, not to mention saying we've humanized the mouse and then wondering why everybody thinks that might be a problem. So we did say, look at language, and then look at how research is overseen and governed and whether there's room for improvement. Here is the acknowledgements of the investigators. Here's a nice picture of, but way back, some of this was done in person, but most of it was done ultimately over Zoom, of course, thanks to the pandemic. And then this is just the beautiful cover by a fantastic artist for our report. And there's a lead article that's fairly long and has a set of 10 recommendations in it that I encourage you to look at and share. And then there are also articles reporting on interviews with scientists and oversight committee members about moral status in chimeric animals that you were involved in, and so that you and that Patty led with Caitlin and you. So that's like empirical research reporting on interviews, and then some essays, including one about, really from two people who are very committed to animal rights and animal welfare, and so we really made sure to investigate and integrate that perspective into the work, as well as we have a lot of people who actually do the science or are involved in the ICOOCs and other institutional mechanisms overviewing the science. So we have these conversations with researchers and animal welfare experts. So that work is available, and we can get into it in the Q&A, but I just want to make sure that I made that really clear that people can find that research. So yeah, I think maybe with that we can just go over to the Q&A. Danny, how do you want us to do that?
I can just offer you some questions, and then whoever feels up to answering it can take the question.
Great.
So we're getting some questions asking for a bit more unpacking on moral status and species membership. Why or why not is using species categorization as a proxy for moral status not ideal?
Okay. That's a good question. So I want to acknowledge that there are people whose entire academic research careers are devoted to the issues around animal welfare and animal rights and this whole debate. And I am not one of those researchers. Like I said, we did have some of those people involved in our study and we learned a huge amount from them. I also, what I understand is that people come to some of the same conclusions from different places on this issue as they do on many other ethics issues that we deal with in our work. So you could have the same view about moral status of a non-human animal, but have different reasons for that view. For some people, I think it is to do with species type. For other people, it might be to do with capacities. For other people, it may be that is a really human-centric and limited way of thinking about what makes for moral status. So I don't even know if it's helpful for me to give my own view on this. I've, like many people, I think I've been kind of it's confronting, but also helpful to think about the ways in which a capacity-based view might be quite limited. So, octopuses have a lot of capacities that we don't, and also don't have capacities that we do. What does that tell us about their moral status? I think that's a very complex question, actually. And one in which in my own life I've had sort of 180-degree kind of change from thinking that maybe because they can't do what we do, they're not high status morally, or then starting to think that that's almost a nonsensical question. So I do think, that's my personal view, I do think that speciesism and species-based moral hierarchies are problematic, but also they are what we use. So I don't, in a lot of, just in law and in regulation. And so, this is what I meant when I said that these debates about chimeric research really do intersect with these much larger shifts and debates in public discourse and individuals' own kind of moral thinking about non-human animals. And raise questions that have nothing to do with chimeric research in a way, which is like, what do you eat? What do you kill, etc. So I think it gets very big very quickly, and that's part of what was really stimulating, honestly, and on a personal level for me, just to be involved in this project. But I can't kind of categorically answer that question. I don't think. I'm not sure anyone can.
Yeah, I think it's a very interesting question. Sort of what's the relationship between moral status and species type. And I think there's so many interesting nuances here to talk about. I don't think saying something has moral status or not gets you very far. I mean, there are people in the animal research committees that'll think that mice have a certain level of moral status. You could wrong them by making them suffer unduly or unnecessarily or extremely. So they say they have some moral status, but it doesn't mean that you can't use them for research. So the more interesting question for me is not whether they have moral status or not, but where that threshold for intervention is, right? So animal research, all the oversight and regulations around it for research institutions kind of is part of the Animal Welfare Act. And that's kind of an acknowledgement that animals have some kind of rights not to be, harmed or unduly suffer. The question is there like a no-go zone, or are there some species for which, no amount of any kind of use is ever justified? And I think that's the more interesting question for me, rather than is there like a species type and a certain level of moral status attached to that. I think it has to do more with thresholds. I think we can generally say all animals that are sentient have, because of their sentience, have some degree of moral claims on us of how we treat them. The other thing I find interesting about species is that you might have a view that a particular species of animal shouldn't be used for research, invasive research purposes. Maybe behavioral research is fine, but invasive research where you're doing operations on them is not permitted. Period. So you can kind of be that person. Another might be, I'm okay with this hypothetical species X being used for invasive research, but if the transfer of human cells gives it capacities that make it extra, like it's no longer equivalent moral status member to its other species, like in the control group. So let's say hypothetically, you have mice in the control group. You have experimental mice, that same strain, same breed, you put the cells in, they somehow have a gain of function, that it would be wrong then to do any further studies on them that involve invasive studies or euthanasia because you somehow elevated them. So that's an interesting line as well, right? A threshold, because that could sort of move. In my experience, I think that the transfer of any kind of modification or cells typically makes them less competent or less species typical. And so you actually see a drop in welfare rather than like gain of function and suddenly, they're enjoying life a little bit more. I mean, it usually is a drop. So that's kind of the more likely outcome. And then you have people who think that there are some species that are so close to human that if you didn't do anything that made them gain function, they would be okay. But like, let's say hypothetically, some non-human primate might get mental capabilities, cognitive capabilities that would kind of bump it up to this zone where it's above the capabilities of the control group, and that's somehow problematic. So there are all these like dimensions to this question of the relationship between species and moral status. I think it's really about thresholds and whether the intervention kind of like raises the threshold, and people have thresholds at different places for intervention. It's a really intriguing question because I don't think because of that, you can't give like one like common answer across the board, because there's all that variance in how people think about go or no go on the experiment.
Yeah, and I just want to acknowledge as well that non-human animals can have different moral status for people with different moral significance based on very different things than their species function or their capacities. So I live in my home again of New Zealand, Aotearoa, and like native bird species have really high significance here, moral significance. That's not about honestly, there are birds that can't fly, right? Like some of them have kind of, you're like, what is the capacity is not the issue. It's to do with their relationship to native culture and native beliefs and systems. They're rare, that's already a thing. So there are many different things that feed into that. We've got a question here about, sorry, Danny, you wanted to read the question? Sorry.
You can also read it, Josephine.
It's just about, I think it's a question that Insoo may be able to address. We had members, it's about whether or not we queried IACUCs to identify whether they assess research using the welfare concept. We did have IACUC members on our expert working group, so we had input at that level, but you did empirical, did interviews involved with interviews. So you want to maybe
right. Yeah. So the Case Western team did a series of qualitative interviews with people who either were doing research as scientists in the Chimera field, or were people part of oversight committees, or were animal research oversight committee members. And so some of these conversations, these interviews were summarized in like this conversations piece that's in our special issue. So moderated by Caitlin Craig, our research assistant. She went through and she had a really interesting conversation, which she then transcribed, and you can read it with IACUC members and their views. And many of these people have actually like reviewed chimeric research protocols and were there to kind of monitor the welfare of the animals. So there's a lot there. I would turn the person who asked that question to that set of conversations or essays in the special issue.
Yeah, and I think they are attuned to the issue of, I think that's fair, right? That they're very attuned to this question of whether or not there might be novel changes. But it's also a tricky question, is what I understood from that research. So it's not, you're already looking at non-human animals in an unusual environment because it's a lab. So I think this question about whether we would expect human cells to have sort of result in novel changes and what those might look like, and novel and unexpected, I guess, as well. So that was like a focus of our study, and definitely something that was a rich kind of vein that we looked at in the work.
Yeah. I do want to say in this webinar something that I actually don't think I've ever like written anywhere, but I can share with you some of this experience. So in my conversations with chimera researchers, when I bring up the issue of, do you monitor the chimeric animals to see if there's any kind of like behavioral changes? And they say, well, to a very limited degree, because they'll look at the control group and they'll look at the experimental group to see if there are any like noticeable differences. But there's a limit to what kinds of tests they'll do, because if it kind of falls outside the aims of the study, if it's kind of meant to be like, okay, so let's say hypothetically, the escrow committee, stem cell committee says, we really want you to assess whether or not there's like this kind of gain in function in the cognitive capacities. And they say, well, all we were trying to do is create an animal model of this like brain disorder. So yeah, the first wave will be using healthy cells. There may be a gain of function, but we're really interested in the disease cells that we're going to put in at step 2 to see if that actually gives you the model of the disease. So for that healthy first wave group, they'll say, we're not like animal behaviorists. We're not people who research that. So if you want that assessment, someone else is going to have to do it. And to really like do that like gain of function assessment, that's a really different kind of study. Like that would have to go under a different IACUC and rationalize in some other way. And then you're going to use more animals, and that's not what we're interested in. So it's really kind of an interesting challenge of like bioethicists might say, look, we need to see if there are these like morally significant changes cognitively. The research team will say, oh, great, who's going to do that? Because that's not part of our study. We are really sticking to the IACUC standards of reduce, refine, keep the number small, only use exactly what you need for your research question. This is not our research question. This is your interest, right? Or the bioethicist interest. And it's like kind of like an interesting sort of like, well, how do we ever get this like assessed? And so I just wanted to raise that observation that's a really interesting rebuttal by the research team. They'll say, look, if that's what you want us to do, we have to somehow put it into our grant application, justify it to our funders that we're going to have this third arm, this other assessment for people who are really interested in like the cognition issue. We're a disease group. And that's why you kind of like practically have a hard time actually getting that information, because if that's true, you're going to actually burn through more animals.
Well, imagine that, like that is like, imagine that you were concerned, and then that's the result was that more non-human animals would be involved in research.
Right. It's kind of
so that's one of those things where you just absolutely have to think very carefully about the implications of various different sort of concerns that you might raise and like suggestions. So we kept coming back to that, like trying to returning to this issue of the sort of preciousness, I guess, or the importance of non-human animals and the challenges and responsibilities around their use in research, and not losing sight of that in our kind of honestly like slightly human-centric kind of set of worries about what this might mean for us, and who we are and where we stand in the hierarchy. So we really did try to keep coming back.
Something also I should point out that people seem to forget this about chimeras is like each chimera model that you make is like a one-off bespoke model. It's not like you're making a new strain. Like if I'm really worried about like the generic chimera research concerns, I would be much more worried about genetically modified animals. that have like that you can start breeding. You can make like a whole new strain of an animal species, monkeys or whatever. That kind of you are creating a new type. The chimeras are just like one at a time, transfer cells. Just because you did it like one time, you do the same protocol, you repeat it, you're not always going to get the same result. And so it's not like you say we have an experiment where we're going to create 10 chimera, neurological models, and then somehow that's going to like change how these animals are going to be indefinitely in the future. It's one at a time, and they don't always kind of end up the same because the cells don't always integrate. There's a lot of variation. So that kind of that worry that I think people are trying to get at, like, are you creating a new morally concerning type of animal? Because I think a little bit less than it's one at a time. It's bespoke. It's not breeding. It's not creating a new strain. If you're really worried about a new type of animal with ambiguous moral status, I think you should really focus on genetic modification.
And I do want to just point readers as well to Carolyn Newhouse's essay in our special report, which talks about some concerns about we noted that there are these animal welfare concerns that are much bigger than chimeric studies, but definitely need to be front and center when thinking about this research. And she also raises and talks about they're not animal welfare concerns, and primarily they're concerns about the rigor and the usefulness of these models and non-human animals as models, and like making and that intersects with this much larger ongoing discussion about some of our regulatory rules requiring non-human animals to be used in clinical studies, for instance. And we saw with COVID that there was an exception made, and then there's a new FDA policy that will not necessarily always require non-human animal experiments before moving into human trials if it's not scientifically required. So there's a set of issues that are about scientific rigor that could also result in fewer non-human animals being used in science out of a concern for like just doing using different things in a dish that can be more helpful. So there's another sort of big debate that intersects with this work as well, that we wanted to bridge in here in our work.
Yeah, and I should point out that the technology that enables these alternatives to animal use come from the stem cell world, or organoids. Or organ chip systems, which the FDA has very recently said they might accept as an alternative to animal testing. So just you wait. You might see that some of the conundrums that were raised by the stem cell field end up being resolved by other kinds of stem cell technologies.
Yeah, great. Danny, is there anything else we should address?
I think we have enough time for just one more question, and this is one that's more broadly about the field of bioethics. Throughout the report and in this webinar, it seems like there's a common thread of understanding this relatively new problem through older questions and through older issues. So I was wondering if you could just talk more broadly about the lessons gleaned from this project and what bioethics can carry forward from it. Specifically, how can the field better learn how to lean on these quote unquote old insights?
That's a great question.
I
was just going to say that I think so one of the things that I think about that you made me think of with that question, I'm not sure if it's a direct answer, is that in academic work, it's very easy to be quite narrow and specialized, and a project like this shows to me that one of the beautiful things about bioethics is that you have to be a little bit of a big picture person who looks across different disciplines, different types of questions, and sees the recurring questions and puts them back together. And that is something that comes from going a little bit wide and not just narrow. And so I do think that, one of the great things, and certainly one of the rewarding things for me as an individual of being in this area, is that you need to go wide sometimes. And that's a very, we live in this ever more specialized kind of universe. And I think that's the thing that I think that, is one of the most exciting and also honestly pretty challenging things about working in this area, but where I think this project somewhat typifies that particular sort of a benefit of the field.
Yeah, when I take a step back and look at sort of our approach in this special report and in our project, I think if it says anything about where bioethics is going, I think to me, it's like very unabashedly heading into the secular bioethics area. So, traditionally people looked at chimera research kind of almost from like a religious or natural law tradition of like it's unnatural to mix species and kind of like, but we were really focusing on harms and benefits and a little bit more of a universal language that people could sort of from different cultures kind of point to and reason out. I think it just underscores when I look at kind of our approach to the chimera issue, the problem of chimeras. It's further along the pathway of like coming up with the reasons that other people could kind of publicly discuss and examine. And I think animal welfare and suffering is kind of a pretty common, like publicly available set of reasons to deliberate about. And I think our approach only reinforces that kind of trend in bioethics to kind of go secular. There are, of course, many other approaches to bioethics that are much more rooted into like, communitarian or kind of more like, traditional value systems that are a little bit more coherent, not so pluralistic, but we're definitely not doing that. So I think that that's apparent to anybody reading the report.
I think we're at time, but thank you so much for joining us today for this webinar, and a special thank you to Dr. Insoo Hyun and Josephine Johnston for speaking on this really interesting and important topic.
Thank you.
Like mentioned, of course, like we mentioned earlier, a recording will be available later today on the Hastings Center website, including closed captioning and other accessibility features.
Thank you so much. Thanks everyone for tuning in.
Thank you, everybody.